Warmest greetings to all my incredible subscribers and new viewers alike. Welcome to our channel, where today's focus is all about HSH. Let's get started. Hematopoietic SH domain containing SH protein is a protein encoded by the hematopoietic SH domain containing SH gene. As we enter this new phase, let's analyze gene from different angles and evaluate its significance. SH is located on chromosome 19 at Common aliases of the gene include hematopoietic SH protein and ALX adapter in lymphocytes of unknown function X. The MNA encodes two main isoforms. Isoform 1, the longest isoform, contains 7 exons. The gene spans from 16,134,028 to 16,158,575. Get ready for an exciting exploration as we unravel the mysteries of MNA. Two main isoforms of SH exist. Isoform 1 has 7 exons and is 2,403 in length. Isoform 2 has 6 exons and is 2,936 longitude. Although isoform 2 has longer MNA, it still produces the smaller isoform in the mature protein. Isoform 2 has a variant 5 UTR and a different start codon, as well as a shorter N-terminus. The MNA has a short 5 UTR and a long 3 UTR. With that being said, let's now move on to protein. The protein has a molecular weight of 39 kilodaltons and a pi of 6.678. The main feature of the protein is the SH homology domain, which is a region that has phosphotyrosine receptors and is important in many signaling molecules. This domain is located from residues 26, 100 and 27. The secondary structure of the protein contains a helical section around residues 40, 50, a sheet between 60, 70, helix between 100, 110, 135, 145, 175, 180, 200, 225. And additional sheets between 235, 240 and 295, 300, shown in the figure at the bottom of the section. Helix are purple arrows and sheets are red arrows. The protein has several locations of post-translational modifications, especially phosphorylation and galactoglycosylation, which has been shown to play a role in cancers. The tertiary structure of the protein has not been confirmed through research. However, predictions using ITASSER software are useful in visualizing the protein. As we progress through this video, let's shift our attention towards expression and uncover its hidden depths. Based on NCBI-GEO expression profiles and EST analyses, the protein appears to be narrowly expressed throughout human tissues. It is highly expressed in bone marrow and T cells, lymph node, mammary gland, spleen, stomach, thyroid, and small intestine tissue. Expression is elevated in cases of early T-cell precursor acute lymphoblastic leukemia and lowered in breast cancer cells that are treated with estrogen, suggesting an interaction between the protein and estrogen. As we transition to the next segment, let's unravel the mysteries surrounding function and gain a fresh perspective. The function of the SH protein is still not fully understood. However, it has been shown to play a role in various cellular functions such as apoptosis, wound healing, vascular endothelial growth factors, membrane-associated intracellular trafficking, beads of lipid droplets, and collagen remodeling. It is also thought to play a role in T-cell activation. In this part of the video, we'll be diving deep into interacting proteins and unraveling its profound impact. SH interacts with several proto-oncogenes, including FES proto-oncogene FES and CRK proto-oncogene CRK. It also has suspected interactions with other proteins such as tyrosine kinase non-receptor 2 TRK, PTEN-induced putative kinase PINK, and interleukin-2. A summary of these proteins is shown below with their suspected functions. Name NCBI accession number function FES proto-oncogene FES NP1000 996.1 hematopoiesis growth factor and cytokine receptor signaling. CRK proto-oncogene CRK NP58431.2 adapter that binds to tyrosine phosphorylate proteins. HSH domains tyrosine kinase non-receptor to NP5772.3 tyrosine kinase, which may be linked to tyrosine phosphorylation signal transduction pathways. PTEN-induced putative kinase pink NP115785.1 threonine protein kinase interleukin-2 NP577.2 cytokine important for T and B cell proliferation. In this section, we'll be exploring clinical significance. The SH2 protein has been studied along other human genes predicted to be involved in the human immune system. SH2 was found to be highly expressed in patients with ulcerative colitis. The protein is also associated with alpha interferon activity. In the next segment, we'll be exploring homology and its implications for our subject matter. SH2 has 4 distant paralegs and several ortholegs in other species that have high levels of conservation. Let's now shift our focus to paralegs and explore the ways in which it shapes our perspective. The 4 paralegs of SH2 in humans are other proteins containing SH2 domains. They do not have a high level of conservation other than this domain. All paralegs were found through genicodes. Name NCBI accession number sequence length amino acids sequence similarity sequence identity SH NP1,154,913.1 399.29% 21.7% SH NP1,094,874.1 451 33.1% 25.9% SH NP1,167,630.1 454 12% 17.4% SH NP997,255.2 357 33.7% 18.1%. Let's now turn our attention to ortholegs and uncover the fascinating insights it brings to the table. SH2 has several ortholegose proteins that span across several orders of species. The protein was well conserved across mammals as well as a few reptiles, amphibians, and invertebrates. The following list is not exhaustive, rather, it shows the wide range of organisms that the protein may be found in. All ortholegose proteins were found with BLAST or BLAT programs. Scientific name, common name, order, NCBI accession number sequence length amino acids sequence identity sequence similarity Pantroliditis chimpanzee primates NP1,229,302.1 350 99% 99.7% heterocephalus glaber naked mole rat rodentia ebb 324 54% 63.4% Hipposideros armida great round leaf bat Chiroptera XP 19,497,370.1 360 67% 76.1% Condylora cristatus stun-nosed mole Sauricomorpha XP 4,688,256.1 355 62% 72.1% Camelus droboreus dromedary camels Separutila XP 10,993,355.1 360 66% 75.7% Pantherpodus leopard carnivora XP 19,271,712.1 360 62% 71.4% Melagris galapodo wild turkey bird XP 10,723,595.1 326 23% 28.8% Anolis carolinensis carolina anole reptile XP 16,854,511.1 567 22% 31.5% Xenopus tropicalis western clawed frog amphibian XP 12,800,000 809,627.1 363 31% 42.8% Carentius milli Australian Vossart fish XP 7 million 899,329.1 526% 33.1% Lingula Anatina Lingula invertebrate XP 13 million 404,014.1 187 18% 23.4% Beyond Valoria Glabratinor invertebrate XP 13 million 80,000 865.1 818 12% 10.1% Salpinga carusidinor protissa XP 4 million 995,081.1 481 17.7% 10.9%. Remember to follow me on social media for behind the scenes content and updates.